water quality parameters, and we will be focusing on biochemical oxygen demand. This topic is very common in wastewater engineering. You have already studied biochemical oxygen demand in detail in your previous course, titled as Advanced Wastewater Engineering. Today, we will just to go through this topic so that you can have a revision and recall of what chemical oxygen demand is and how it is applied for wastewater engineering. So, furthermore, our also target for discussing chemical oxygen, that we can understand what are the major constituents present in our wastewater sources, what are their concentration, and in what proportion these organic content are present in our wastewater, so that we can understand the processes which are actually responsible for their degradation and for their treatment. So starting with the organic constituents, if you see the organic matter in wastewater, it consists of proteins about 40 to 60%, carbohydrates about 25 to 50%, and oil and fat about 8 to 12%. Urea is also one of the constituents and major constituents which is present in wastewaters. It is available only in fresh wastewater because it decomposes rapidly, and that's why it will no more be found in wastewater later on stages. So, measurement of organic content, we need to have organic content measurement methods. So, biochemical oxygen demand (BOD test), COD test, chemical organic theoretical oxygen demand, which is calculated on the basis of chemical formula of organic matter. So these are the methods and calculations which help us in determining the total amount of organic content present in the wastewater. And it is to determine approximate quantity of oxygen that will be required to biologically stabilize the organic matter present, and to determine the size of wastewater treatment facilities, to measure the efficiency of treatment, to determine compliance with wastewater permits. Very simple, go through. And then, what are the bases for biochemical oxygen demand? If sufficient oxygen is available, aerobic biological decomposition will continue until all the waste is decomposed. So this is major thing of the biodegradation. As long as we have oxygen available, most of the time aerobic microorganisms will dominate. They will lead the process of degradation. But gradually, as you can observe in our wastewater engineering, gradually the biodegradation shifts earlier from aerobic. Then what is the reason to move to anaerobic? It is the deficiency in oxygen available. Initially, we have very high levels of saturation of oxygen, particularly at the surface, because it is directly in contact with the source, that is atmosphere. But gradually you will find that there will be a layer of the microorganisms developed with the passage of time on the surface, and because of that the movement of the oxygen to the lower layers will be abundant will not be available. So in that case, in the lower layers particularly, not in at the surface, but at the comparatively deeper layers, there is no oxygen and there is no availability of light. So what will happen? Anaerobic microorganisms take lead and they start degradation. So that's the second category of treatment. So starting with the very simple parts, if we have sufficient oxygen, aerobic biological decomposition will continue until the waste is decomposed. Then a portion of the waste is oxidized to end products to obtain energy for cell maintenance and formation of new cell tissue. Very much understandable because when we have the organic content with us, uh, it will decompose into what? CO2, H2O, and release of energy. This is the equation. So what will happen? That energy is obtained and that energy will be again required by the microorganisms which are flourishing, which are growing in that environment. So it is sufficiently meeting the needs. Then simultaneously some of the waste is converted to cell tissue using part of energy released during oxidation. Now, this is important. Cell tissue, what is cell tissue? Uh, and see on the left side of the slide, tissue is intermediate level between cell and complete organ. Tissue is an ensemble of similar cells from the same origin that together carry out same function. Organs are then formed by functional grouping together of multiple tissues and then the whole structure develops. So, simplifying the situation, the waste is being converted into CO2 plus H2 and then energy, and that energy is being used in developing cell tissues. What are cell tissues? These are the body organs, unit of body organs, which are having same kind of cells. Now, because they have same kind of cells, so they are performing same function. So tissue is forming one at a time with the combination of different cells. Okay, then finally, when waste is used up, new cells begin to consume cell tissue to begin to obtain energy for cell maintenance. Now, this is the stage when you understand the logarithmic growth of microorganisms. So you need to understand the growth curve of microorganisms, which is very basics of wastewater engineering. How the cell grows in an environment. So if you consider the growth curve, it consists of four phases. What are those four phases? First of all is the acclimatization phase. Then it is the growing phase, which is normally logarithmic phase, in which growth is exponential. Then we have a stationary phase when food is depleting and we are having equal number of dying and growing microorganisms. And then we have a decline phase in which there is death of microorganisms. Same is the criteria here. Now, in the context of cell growth, uh, cell growth curve, please read the last line. Finally, when waste is used up, now the food has ended, new cells begin to consume cell tissue to obtain energy for cell maintenance. That means it is hindering the growth of new microorganisms. So at this level, we are going to complete our growth curve. So this is called endogenous respiration. So this is if you have a view here, you can see how we will be converting the organic content into the simplified form, and then you can observe that how cell tissue is being formed, and this is the a better version of the same equation: oxidation, synthesis, endogenous respiration process. So these three equations are going to demonstrate the whole process of the wastewater degradation process. This is our fundamental basic formula. It's actually not formula but a symbolic formula representing the wastewater, because it is showing all the ingredients: uh, carbon, oxygen, hydrogen, nitrogen, sulfur, and in the presence of oxygen and the presence of microorganisms, bacteria, they are producing CO2, H2O, ammonia, other end products along with energy. Now, what is being synthesized in this case? So, if you find out, we are using this energy in the presence of oxygen used by the bacteria to produce new cell tissue. Now, this new cell tissue is being produced. Again, the bacteria are using the energy obtained from the decomposition of the organic content. This one. Now, what will happen? That this new cell tissue will again be consumed, and it will be giving CO2, ammonia, and 2H2O, oxygen, water. So this is called endogenous respiration, in which new cell tissue is again being consumed. This is kind of reduction process. So if only the oxidation of organic carbon that is present in the waste is considered, the ultimate BOD is the oxygen required to complete the three reactions given above. This oxygen demand is known as the ultimate carbonaceous or first stage BOD, and is usually denoted as UBOD, ultimate BOD. Now, this is the time to understand ultimate. Actually, what BOD is? BOD is the demand. Actually, it is measure how much organic content is present. Higher the organic content, higher the oxygen demand. So link it with the UBOD, ultimate BOD. So ultimate BOD is the combination of all the requirements of oxygen. This is the requirement. This is the oxygen requirement. This is another oxygen requirement. So combining all these requirements is going to describe the overall oxygen requirement for the final decomposition of the organic content. That is leading us to a term called ultimate BOD. So on one side with ultimate BOD, which is supposed to be considered as the total organic content or total waste present in the sample of wastewater. So just keep it in your mind while we are going to while we will be describing some other terminologies as well in our coming slides, and those slides we will be discussing about other similar terminologies like UBOD. What are other like remaining BOD, remaining BOD exerted. So how to differentiate between these terminologies? Well, moving forward, so I think we have talked about it. Okay, so this is the determining procedure, and BOD is actually the oxygen, dissolved oxygen level immediately after preparation. U2 is the dissolved oxygen of diluted after 5 days at 20 degrees. So keeping it simple, BOD is actually the demand of oxygen being used, and the start of the experiment until the end of the experiment. for between these two timings, we will find out how much oxygen is consumed. Then we will directly link it with the reaction rate. Then we can find out different terminologies related to BOD. But simply BOD is the difference in time, in difference in oxygen consumption, oxygen saturation at the start of the experiment until 5 days. BOD test takes 5 days. So this is a question: why 5 days? So we will try to answer it later on. Just immediately the answer is that we are only focusing on the carbonaceous BOD, which only deals with the initial 5 days. And after 5 days, the nitrogenous BOD starts in its role, and then nitrogen starts decomposing, and then we will be more focused with the overall BOD, and it can lead to 20 days test. And then during these 20 days, all of the 99% of the organic content will be decomposed. So up to 5 days, we can get a normal trend. We can get rate at which the BOD is being consumed, oxygen is being consumed, and then we can determine the organic content from the equation of our BOD kinetics. We will be shortly talking about the BOD kinetics. Initially, just to have a concept, what is this? It will be showing the rate of decomposition, so the organic content. So all is written here as well. Biochemical oxidation practically takes infinite time to go to completion because the rate of oxidation is assumed to be proportional to the amount of organic matter remaining. This is the basics of the kinetics. So within a 20-day period, the oxidation of carbonaceous organic content matter is about 95 to 99% complete, and in 5 days period used for BOD test, oxidation is around 60 to 70% complete. That's the story. That's the summary of the BOD. Again, it's the definition. Go through it, please. So it is performed for the streams and the rivers because huge amount of wastewater is added up into our rivers and drains on daily basis. It contains huge amount of organic content. So we need to determine how much water is being added up. So when our water is in huge quantities, we need to dilute it because we must continue the process. Oxygen once saturated in the bottles of BOD must sustain up to 5 days. But if the organic content is high, then all the oxygen within the jar or bottles will finish within 1 day or 2 days. So it will not give us a precise oxygen consumption rate because it is not completing in 5 days, but it ended up in 1 day only. So we must sustain the oxygen saturation up to 5 days. How can we do it? By reducing the level of organic content. So we will prepare different dilutions and we will see which dilution works, and then we will make that dilution use for that dilution for our experiment on BOD. Then we will multiply the answer by the factor of our dilution so that we can get the original value of the organic content. This is the summary. I think you already know. We are now adding up a factor which is called as dilution factor, and we will be then uh, minusing it from our original oxygen levels at zero time, and then this is actually we are also adding up the dilution factor, so it will give us the BOD 5. I think it's quite clear to you already. We are just revising it, so we need not to spend more time. But if there is still some ambiguity, on, ask me any question that will be clarified. This is important now. Nature of BOD reaction. How BOD reaction takes place. What is the relationship between the organic content and the time of its decomposition? Very simply, very simple. Higher the organic content, higher will be the rate. Of course, when there is more food, there is a faster reaction. But gradually, with the decrease in the organic content, the rate will slow down. This is actually a reduction with the time. That's why we will be using minus sign with it. So try to read it here. The rate of reaction is proportional to the amount of oxidizable organic matter remaining at any time t. So mathematically it's like this, in which C is the concentration of oxidizable organic matter at a particular time t, and then again the concentration uh, multiplied by the constant. So this is the differential. So the amount of decomposition with time will be giving us the concentration of the organic content itself. Just for our ease, replace this C with L. So now this L sign is used for BOD value, and it is shown graphically as follows. This is the ultimate BOD. Ultimate BOD, as we talked earlier, it is the oxygen consumption in all the processes. So it must be defining the total content of waste. So at only at time zero we can have the total waste, because with time it will start depleting. So when we will have maximum time, maximum of waste, it's only at time zero. So at time zero we will be having the maximum level. Please go through this equation where K is the BOD reaction rate constant, or biochemical oxidation rate constant. Well, value of K varies with time of waste. T is the organic matter remaining after. This is remaining. Okay. And LT over L is the remaining fraction of the oxidizable organic matter. So if this is the organic matter still remaining, and this is the total BOD level. So if we are taking the ratio of both, it will be giving us which the amount of waste is which has been still remaining. This will be giving us the remaining fraction. Is the remaining fraction of the oxidizable organic matter. And if we multiply is this ratio from 1, it will give us the amount oxidized which has been done. So this is the remaining fraction. This was the overall fraction L value, and the ratio will is giving us remaining fraction. So if we minus it from 1, it will give us the fraction oxidized in time t. Now the total fraction oxidized in T days is obtained by multiplying it by the total amount of organic content L. If Yt is the amount oxidized in T days, then this is the equation. So Yt is equal to 1 minus Lt over L multiplied by L. BOD exerted with time is shown graphically here. So this is the BOD oxygen demand on this side. We are having maximum demand at this level, and gradually the BOD remaining is slowing down until it reaches the minimum value. On the other hand, BOD exerted is the amount of oxygen consumed. So initially there was no oxygen consumed, and when we started the experiment, the oxygen started consuming, and it's all about the oxygen consumed. Now there is a relation. Amount of oxygen consumed is going to give us the total organic content. So please link, you can link it with. Now this is again a brief description and kind of repetition of the previous slides, but maybe it will explain the concept more easily. The rate of oxidation exertion is modeled based on the assumption that amount of organic material remaining at any time is governed by first order function as given below. So this is the amount remaining. Earlier we set it DC, and this is the derivative with reference to T. This is the BOD remaining. Earlier we talked it about C, and because these are C, and then we replace them with N. So BODR is the first derivative of the UBOD, and if we are going to integrate it, we will be getting this equation. We have earlier seen the derivation, please check it. And then we can have the time limit starting from 0 to t, and then we can further express BODR is the amount remaining at time t, K is the first order reaction constant, rate constant, UBOD is the total ultimate, or in this case BOD, T is the time. So BOD exerted up to time t is given by, this is the amount exerted. So here it will be like UBOD minus BODR. This is YT in our previous sample of equation, and BODR is here expressed in terms of ultimate BOD as the first reaction rate constant of integration. We get this equation, and now we taking it common, we can express this equation like this. So this is almost the same equation. Here we talked about the expressed it in terms of YT, now it is BODT. So it should be noted that in literature dealing with the characterization of wastewater, the term L and BODU are often used to denote ultimate BOD. So please keep it in your mind. And then K1 is the untreated wastewater value, and it actually depends on the temperature as well. We have a graph for values. You can get the values from there based on. So again, it's if you want to convert this K in base 10 form, you can use this reaction. It's kind of detailed which you can go through by yourself. Please, I will request you to go through these examples. It's very important, and it will clarify concepts about BOD, ultimate BOD. I'm more interested in ultimate BOD. Please clarify what is ultimate BOD. And then YT is also important. In other words, we talked about it in BODT. What is BODT? Please check it. Two important things, ultimate BOD and SOID. Please go through these examples. It will clarify your concepts. Well, now we will move forward and we will talk about nitrification test. Well, earlier we were only talking about the carbonaceous peak. Now we will move forward to nitrogenous BOD. So, non-carbonaceous matter such as ammonia, which is produced during the hydrolysis of protein, known as bacteria are capable of oxidizing ammonia to nitrite and then to nitrate. Nitrite is NO2 and then nitrate is NO3 ions. So here, if you see here, conversion of ammonia to nitrite by Nitrosomonas bacteria, NH3 is being reduced to this nitrite. Now conversion from this nitrite to nitrate is nitrobacter. If you see here, this reduction will lead to the HNO3. The overall conversion of ammonia to nitrate is given this way. So um, the reproductive rate of nitrifying bacteria is slow, and it normally takes 6 to 10 days for them to reach a significant number to accept measurable oxygen demand. However, if a sufficient number of nitrifying bacteria is present initially, the interference caused by nitrifying can be significant. So let's go through it. And this is a typical expression. If you see the curve of BOD test, we will be having this kind of behavior. This is oxygen consumption, and after 5 days, we will be having an ascent because of the nitrogen biochemical oxygen demand. However, carbonaceous is also continuous because we have only consumed about 60 to 70% of in the first 5 days. We were only interested to get the rate, and now actually it will be depleting the carbonaceous organic content to 95 to 99% in 20 days. And meanwhile, at the same time, the nitrogenous BOD is also active. Now, nitrogenous compounds and all nitrogenous material is also being decomposed. So, now this is separately describing carbonaceous BOD, and then please go through these contents. These are just explanations theoretically. So, this is theoretical oxygen demand. What is theoretical oxygen demand? It is the calculation of oxygen demand by using the formula. If we have a balanced equation, we can easily find out how much oxygen will be consumed by the microorganisms. So if we have all the molarities and we know the molecular weight and we know exactly what is present in the wastewater, then we can easily develop such equations and we can find out the actual consumption. And at the same time, we can find how much oxygen has been consumed. Similarly, for the nitrogenous BOD, we have these kind of predictions which can determine the oxygen consumption. Then the theoretical oxygen demand can be determined by using this method, and this method is actually used to confirm our experimental work conducted in our lab BOD test. So again, same thing. Please go through these examples for practicing and for clarifying your concepts. So this is the again a theoretical representation of BOD as a function of time. So this is the BOD and heading towards ultimate BOD, and then at 5 days BOD you can see this is the situation, and then on the 6th day the nitrogenous BOD has also started. So this is again the definition of ultimate BOD. Go through it. Uh, kind of explanation we have already talked about. Oh, this is another explanation. Please let's look at it. See here, this is the ultimate BOD to this level, and this curve is giving us the amount of organic waste remaining. It is quite depleting fastly remaining. And at the same time, you see here the total amount of organic matter remaining in BOD bottle. This is due to the waste, and this is due to the total organic content. Now, what is the difference? In this one, we are also adding a portion of the dead microorganism, which was explained earlier in terms of endogenous respiration, if you can recall. And then it is the oxygen consumed for the development of cell tissues, and then those cell tissues will be decomposed again when any will be finished. So this is the difference. Is it clear? And then it will exist here, and you can see this is the BOD remaining. This is the BOD exerted. How much BOD has been exerted? This is the equation going to give us that value. This is lag phase, log phase, stationary phase. This is the depth. This is something about the growth curve we talked about, because the microorganisms first of all move forward to the lag phase. This lag phase is actually acclimatization phase. The microorganisms are getting into a new system, a new environment. First of all, they need to acclimatize. First of all, they need to fulfill their requirements. They need to have their nutritional requirements there. So once they are confirmed the availability of all the nutritional and environmental conditions available in the system, then they will start growing. They will start uh, using that food and that environment, and then they will grow in this speed logarithmically, exponentially, unless and until the whole organic content will be no more available. Then they will be all depending upon the this one was the endogenous respiration, the cell tissue matter. So now the cell tissue matter is being decomposed here, and they are depending, the bacteria are depending upon the cell tissue now at this level. And then finally that will also end up, and then there will be a decline in the presence of the microorganism. Now, these are the dead microorganisms that are making the major portion of this organic content in the bottle, and then it will continue uh, as minimum as possible. So please go through this graph. It's self-explanatory, very important to clarify the concepts. Now this is the biodegradation. What kind of biodegradation is possible? And if we talk only about aerobic biodegradation, simplifying the things, we are actually having organics, complex, presence of oxygen, they are being converted into stable compounds like CO2 and H2O, along with compound energy. Sulfur will be reduced to sulfate, phosphorus to phosphate, organic nitrogen to ammonia, and then nitrite and nitrate. This is the reduction process. And in anaerobic biodegradation, complex organics will be producing methane gas in addition to the other CO and stable, partially stable compounds. This is the so the nitrogen in protein and urea is organic nitrogen. Organic form include nitrate, nitrite, nitrogen gas. Organic nitrogen is found in the cells of all the living things. So let's go through it. Now we have four classifications described here. Ammonification. What is ammonification? We talked earlier. It is the formation of ammonium and then furthermore it will be decomposed. So first step is it is actually the nitrogen cycle. So many of the transformations of nitrogen are mediated by bacteria that use different forms of nitrogen fuel. Some of their metabolic processes. During the processes of decomposition, the nitrogen protein is transformed eventually to ammonium, a certain bacteria. This process is called ammonification. So organic nitrogen being converted to ammonium nitrogen is called ammonification. And this is conducted by nitrifying bacteria. So what is ammonification? It is the organic nitrogen being converted into ammonium nitrogen. Ammonium nitrogen will again be converted to ammonium and protein transformed to ammonium, a certain bacteria. We have nitrification. So now other kinds of bacteria change this ammonia, which we have already seen, nitrite, and for and then from this nitrite it will be converted to nitrate, and this process is called nitrification. It's an aerobic process. Uh, Dead nitrogenous organic matter and release the nitrates. Initially, the ammonia is formed, which react with substances like calcium carbonate. Nitrosomonas bacteria further change nitrite to nitrate. So this is ammonium nitrogen being converted to nitrite and then nitrate. These are the bacteria responsible. So this was the second step. First was the ammonification, now nitrification. Now denitrification. Nitrite, nitrate again being converted to nitrogen gas. Other bacterial species, so it is an anaerobic process with no oxygen, low concentration of oxygen available, leaching field soils. So just you can understand that the first of all we produced ammonia in ammonification, then nitrate in nitrification, now nitrogen gas in denitrification. From this nitrate, now nitrogen fixation. This nitrogen which was produced in the uh, phase will be converted into organic nitrogen. Now, this is a very difficult thing to convert the atmospheric nitrogen into organic nitrogen. How it will be done? It will be done through blue-green algae or cyanobacteria. This is the same thing. It can be named in two ways: blue-green algae as well as cyanobacteria. This is the complete natural cycle. If you go through it, you can find all the processes here. So first of all, we have nitrogen fixation, taking the oxygen from the atmosphere, converting it into the organic nitrogen. Of this organic nitrogen being converted to ammonium. By the ammonification from this ammonia, we will be forming nitrate. By nitrification, this nitrate will be converting into nitrogen or atmospheric nitrogen by denitrification. Complete nitrogen cycle, very important and very easy to understand, and it is in the basics of BOD, wastewater engineering. So it's kind of repetition. Sorry for that. Please go through this content as much as possible. And again, processes where these microorganisms are playing their role. We have activated sludge process here, you can see, and then aeration before sedimentation to stream, and then effluent. So this is kind of whole process with different steps. You can go through it by yourself. I think it's quite easy now. The trickling filter and how it is actually working. It's like drum with biofilm uh, fixed on it, and microorganisms are grown up here, and the wastewater comes in contact with this drum, it will start its process of decomposition. So this is kind of uh, a rotatory method trickling filter. So this is again then septic tanks, suspended growth system. This is suspended in which activated sludge process and facultative ponds are the example. Earlier we talked about the attached growth system, in which we talked about the uh, trickling filter. So this is facultative lagoon interaction. Again, very interesting if you find it here. On the top layer of the stream, you can see the oxygen is entering, CO2 is the one entering here, H2S is being released from it. In the presence of oxygen in the daytime, algae will grow, algae will start its process. Uh, and if you see, this release of oxygen is uh, during daytime is due to the release of oxygen from this algae. Algae is a plant, it is going to release oxygen during daytime. So when algae will be present here, it will be uh, releasing oxygen to the environment. At the same time, it will be at night. It will be again disposing CO2, and at daytime CO2 will be taken by this algae, and at the presence of this CO2, this ammonia and phosphates will be used as for algae, and then new cells will be formed, bacteria will be formed. So all these are the different layer process. So facultative sludge lagoon is here, anaerobic zone, and then facultative zone, and then aerobic zone. So then we have this another uh, flowchart diagram. I think you can go through it. It is ASP, activated sludge process. You can go through it. These are the flocks, and this is the mechanism how the returning activated sludge can be quantified. This is the amount, and this is the microorganisms level. Then this is the oxidate. This is the waste load, and then it will be again entered into this aeration tank after having activated microorganisms being added up into this sludge. Again, there will be some quantity of microorganisms present in the form of this XR. Then some other pollutants like oil and grease surfactants, individual organic compounds, water refractory organic compounds, water priority pollutants, all these things you can go through. Priority pollutants, which are going to have carcinogenicity, mutagenicity, teratogenicity, high acute toxicity, organic priority pollutants, all these things you can go through yourself. So please go through these slides and hope it will be very informative for you. And